So the next topic to cover in the renal system is a system within the kidneys called the medullary countercurrent system. This medullary countercurrent system contains a set of nephron structures that work together to accomplish one very important task of the kidney, and that's to vary the concentration of urine. So we talked about this a little bit in class, that we can produce urine of different concentrations depending on our need to conserve water and volume. Okay, so the kidneys can produce urine that ranges in osmolarity or concentration from as little as 100 milliosmoles to as concentrated as 1200 milliosmoles. Okay, and to give you a perspective, isotonic osmolarity, so the osmolarity of plasma and fluids throughout our body, is 300 milliosmoles. So we can produce urine that is as dilute as 100 milliosmoles and is as concentrated as 1200 milliosmoles. So when the body is in perfect balance, meaning we're perfectly hydrated, our plasma osmolarity is normal, we will produce urine at a rate of about 1 milliliter per minute, and the urine will be isotonic, 300 milliosmoles. If the body is overhydrated, okay, the maximal rate of urine production that we can produce is about 25 mils per minute, and the urine can be as hypotonic as 100 milliosmoles. So that's the best we can do in terms of overhydration if we need to lose an excess of fluid and water. If the body is dehydrated, okay, we cannot stop producing urine. Okay? Assuming normal, healthy renal function, no matter how dehydrated the body is, we still have to produce urine because we still have to eliminate waste, and we cannot urinate crystals. We have to dilute the waste in a minimal amount of water. So if the body is dehydrated, urine can get as low as 0.3 mils per minute and as hypertonic as 1200 milliosmoles. But we have to produce urine, which means that we have to lose water no matter how dehydrated we are. Now, this minimal amount of water loss that we have to maintain is what we call the obligatory water loss. Now, because we have this obligatory water loss, our kidneys are actually much better at dealing with persistent overhydration than we are at persistent dehydration. Okay. Persistent overhydration, eventually we will catch up. Eventually we will be rid of this extra water and volume. But with persistent dehydration, because we have to lose urine, the dehydration is only going to get worse and worse. Okay, so that's an important thing to remember. Okay, so the medullary countercurrent system plays a key role in this ability to produce urine of varying concentrations. So the medullary countercurrent system is a key component of this system is an osmotic gradient that exists in the renal medulla. Okay. So if we take a look at this for a moment, if I skip to this slide, the osmotic gradient that I'm talking about in the renal medulla is illustrated here. That in the outside of the kidney, that's the cortex. If we look at the interstitial space of the renal cortex, it's isotonic, right? Just like it is everywhere else in the body. But if we look at the inner portion, the medulla of the kidney, the interstitial space, it has an osmotic gradient. Okay? It goes from 300 and it gets higher and higher and higher as we get closer to the renal pelvis. Okay? And the portion of the renal medulla closest to the renal pelvis has an osmolarity, a maximal osmolarity of 1200. So this osmotic gradient that we see in the renal medulla persists no matter what's happening. It's a standing osmotic gradient. And it's this standing osmotic gradient that allows us to vary how much water is reabsorbed. Okay? We can reabsorb a little or we can reabsorb a lot and produce urine of varying concentrations. So this medullary osmotic gradient plays a key role in the ability to produce urine of varying concentrations. It's created by a unique anatomical arrangement, and this anatomical arrangement is a set of anatomical parts that make up the medullary countercurrent system. Okay? One set of parts creates the gradient. Okay? So one set of structures in the countercurrent system creates the gradient, and then another set actually uses that gradient to produce urine of varying concentration. So let's look at what those structures are. So if you recall, we talked about the two different kinds of nephrons, the cortical nephrons and the juxtamedullary nephrons. And I told you that the juxtamedullary nephrons, with their very long loops of Henle, play a key role in the ability to vary the concentration of water. So these two parts of the countercurrent system include the loops of Henle of the juxtamedullary nephrons. The long loops of Henle of the juxtamedullary nephrons are responsible for establishing, for creating this osmotic gradient in the medulla. Okay? So those juxtamedullary nephrons with their really, really long loops of Henle are the ones that are actually creating and maintaining this osmotic gradient in the renal medulla. Okay? Now, I told you there's another set of parts that use this gradient to vary the concentration of urine. The parts that use the gradient are the collecting ducts of all the nephrons. Okay? Remember, the renal medulla is arranged in these renal pyramids that are a collection or a condensed area with lots of collecting ducts. Well, those collecting ducts, as they move through the medulla, can use this osmotic gradient to vary the concentration of urine. Okay? So the long loops of Henle of the juxtamedullary nephrons establish the gradient, and the collecting ducts of all the nephrons use the gradient to vary the concentration of water. So we're not going to get too bogged down by this, but I told you that the long loops of Henle of those juxtamedullary nephrons are what's establishing this osmotic gradient. Okay. Now, the way that these loops of Henle establish the osmotic gradient has to do with the fact that the descending loop of Henle is permeable to water but does not allow for sodium reabsorption. And the ascending loop of Henle actively pumps sodium out of the tubule. Okay. So this sodium that gets pumped into the interstitial space of the renal medulla, okay, this sodium is what's actively increasing the osmolarity of this interstitial space. Okay. So urine flows down the descending loop of Henle, and then it flows up, and sodium is actively pumped out, and it raises the osmolarity of the interstitial space in the medulla, and it creates that osmotic gradient. Okay. And now, because sodium is actively pumped out, okay, the urine that leaves the loops of Henle end up having an osmolarity that is very, very low. Okay. So the urine, remember, as it's moving up the ascending loop of Henle, it's getting all of its sodium pumped out, so that the urine that leaves the ascending loop of Henle and enters the distal tubule has an osmolarity of 100 milliosmoles. Okay? So If you remember, this number is familiar to us. I just told you that the body can produce urine that is as dilute as 100 milliosmoles, right? That has an osmolarity as little as 100 milliosmoles. So that means that the urine that comes off this loop of Henle is very dilute. And if we don't reabsorb any more water in the distal tubule and the collecting duct, that urine is going to leave the kidneys at an osmolarity of 100 milliosmoles. Okay? And then the most concentrated urine we can produce is actually 1200. Okay, is actually 1200. So notice that 1200 is the maximal osmolarity of the medullary interstitial space. So that means that this urine coming off of the loop of Henle that has an osmolarity of 100, it enters the distal tubule and it passes through the collecting duct. And as it moves through the rest of the tubule, water is reabsorbed maximally. So it enters the distal tubule and water gets reabsorbed until the urine equals 300, right? It equalizes with the osmolarity that's around it. And then it moves through the medulla and more water gets reabsorbed, and the urine as it's moving through is getting more and more concentrated, matching the osmolarity of the interstitial space, until finally it equilibrates with the maximal osmolarity of 1200 just before it leaves the kidneys. Okay, or just before it enters that renal pelvis and leaves the kidneys. Okay, so those two values, the minimal osmolarity of 100 represents no water reabsorption in the distal tubule and the collecting duct, and that maximal osmolarity of 1200 represents maximal water reabsorption in the distal tubule and the collecting duct. Okay. And then I also told you that if the body is in perfect balance, it's perfectly hydrated, urine will have an osmolarity of 300. Okay. Which means that the osmolarity of 300 is the osmolarity of the distal tubule. So under normal circumstances, urine leaves the loop of Henle and water is reabsorbed at the level of the distal tubule. And then no reabsorption happens at the collecting duct. Okay, and then the urine that leaves the kidney has an osmolarity of 300. Okay, so the medullary countercurrent system involves both the osmotic gradient, which is established by the long loops of Henle of the juxtamedullary nephrons, and the collecting ducts of all the nephrons, which use the medullary osmotic gradient to produce urine of varying concentrations. Okay. Now, the degree of water that is reabsorbed at the distal tubule and the collecting duct is determined by the hormone, antidiuretic hormone. ADH determines the amount of water that's reabsorbed at the distal tubule and the collecting duct, because ADH determines the degree of permeability to water of the distal tubule and the collecting duct. So let's look at what that looks like. So vasopressin, or antidiuretic hormone, is a hormone produced up by the hypothalamus and released at the posterior pituitary. And it travels in the bloodstream and it binds to tubular epithelial cells on both the distal tubule and the collecting duct. Okay. When it binds to its receptor on those cells, it acts through a second messenger system, elevating levels of cyclic AMP and increasing the insertion of luminal aquaporins. Okay. If you remember, the availability of luminal aquaporins was one of those factors that determines the permeability of any segment of the nephron to water. So vasopressin binding to its receptor on the cells in the collecting duct and distal tubule enhance the insertion of the luminal aquaporins so water can cross the luminal membrane and then it can cross the basolateral membrane and be reabsorbed. Okay, so if we just back up right here, when ADH levels are at normal levels, so that means that we're producing urine of normal concentration, right, of 300. That means that ADH concentration is at a level that allows for enough water to be reabsorbed to equilibrate to 300. Okay. When ADH levels rise because we need to conserve more water, it increases the permeability of more of the collecting duct to water. Right? We get more of these luminal aquaporins and more water can be reabsorbed, and it equilibrates to a higher and higher concentration. Okay? Until we have maximal concentration of ADH, which allows all of the distal tubule in the collecting duct to be permeable to water, and we're producing urine of a concentration of 1200. Okay? So this is a form of water reabsorption that is independent of sodium. We are not causing enhanced sodium reabsorption when ADH acts on the distal tubule in the collecting duct. Okay? We're only bringing about a reabsorption of water independent of sodium. So keep that in mind.